Tooth defects and loss are common issues affecting millions worldwide, leading to pain, difficulty eating, and a decline in quality of life. Traditional treatments like fillings, crowns, and implants have helped, but they often fall short of fully restoring natural function. As dental science advances, researchers are now focusing on regenerating entire teeth, aiming to provide solutions that go beyond artificial replacements. This new frontier in oral medicine holds the promise of natural living teeth that can sense, remodel, and function just like the originals. Let's explore how tooth regeneration is reshaping the future of dentistry. Tooth regeneration is an emerging biomedical field that aims to regrow natural living teeth and their supporting tissues using a patient's own stem cells. Unlike traditional dentistry, which relies on artificial materials, this approach seeks to restore biological function. Regenerated teeth can sense pain, remodel naturally, and integrate seamlessly with the jawbone and gums. The ultimate goal is to shift from artificial restoration to biological restoration, offering patients a truly natural solution for tooth loss and damage. Successful tooth regeneration relies on 4 key components: suitable cell sources, scaffolds to guide tissue formation, growth factors to direct differentiation and angiogenesis, and an appropriate culture or incubation system. Each element plays a critical role. Stem cells provide the raw material. Scaffolds offer structure. Growth factors signal cells to develop into specific tissues. And incubation systems create the right environment for growth. Only by combining these elements can researchers hope to achieve complete, functional tooth regeneration. Several types of stem cells are under investigation for tooth regeneration. Induced pluripotent stem cells, iPSCs, have high differentiation potential and can be patient-specific, but they pose risks like tumor formation and are difficult to direct into dental cell types. Mesenchymal stem cells, MSCs, are proven for pulp and dentin regeneration and have immunomodulatory properties, but they struggle to form enamel-producing cells. Dental-derived stem cells are naturally predisposed to regenerate dental tissues, yet their availability depends on donor health and age. Endogenous stem cell homing avoids transplantation, but yields inconsistent results. Each source has unique strengths and limitations. Scaffolds are 3-dimensional biomaterials that provide the structure for new tissue growth. They must be porous, biocompatible, and degrade at a rate that matches tissue formation. Types include injectable hydrogels for minimally invasive delivery, electrospun scaffolds that mimic the extracellular matrix, blood-printed scaffolds for precise architecture, and decellularized tooth scaffolds that retain native cues. Synthetic polymers and bioceramics offer tunable properties, while nanomaterials and biopolymers enhance cell interactions. The right scaffold is essential for guiding stem cells into forming functional dental tissues. Growth factors are signaling molecules that guide stem cells to differentiate, proliferate, and organize into specific dental tissues. Key growth factors in tooth regeneration include PDGF for cell recruitment and angiogenesis, TGF-beta for proliferation and matrix production, BMPs for odontogenic differentiation, VEGF for blood vessel formation, FGFs for cell migration and repair, IGF for cell growth, NGF for nerve regeneration, and SDF1 for stem cell homing. Delivering these factors in a controlled, spatiotemporal manner is crucial for successful tissue engineering. Incubation systems provide the environment necessary for stem cells and scaffolds to develop into dental tissues. 2-dimensional cultures are useful for basic studies but lack complexity. 3-dimensional cultures better mimic natural tissue architecture and cell interactions, making them essential for studying mineralized tissues. In vivo animal models are used to test vascularization, innervation, and integration, with several successful demonstrations in preclinical studies. The right incubation system is vital for translating laboratory advances into clinical reality. Tooth development is a complex process that begins in the embryo and involves a series of well-orchestrated stages. It starts with the dental lamina, a band of epithelial tissue, which forms a U-shaped band along the future dental arches. The tooth germ, derived from ectoderm and ectomesenchyme, organizes into three parts: the enamel organ, dental papilla, and dental sac. These structures give rise to the enamel, dentin, pulp, cementum, alveolar bone, and periodontal ligament. Understanding these stages is key to replicating tooth formation in the lab. Tooth development proceeds through distinct physiological phases: initiation, proliferation, morphodifferentiation, histodifferentiation, and apposition. Morphologically, it passes through the bud, cap, and bell stages. Each stage is characterized by specific cellular arrangements and activities. For example, the bud stage features a small cluster of cells, the cap stage introduces the enamel organ, and the bell stage sees the formation of key layers and the beginning of tissue differentiation. These phases are critical for proper tooth formation and are closely studied in regenerative research. The bud stage marks the initial aggregation of cells that will form the tooth. As development progresses to the cap stage, the enamel organ forms a cap-like structure over the dental papilla, and the dental follicle surrounds both. The bell stage is divided into early and advanced phases, where the enamel organ takes on a bell shape and differentiates into 4 key layers. These stages set the foundation for the formation of enamel, dentin, pulp, and supporting structures, and are central to understanding how to engineer new teeth. During the cap and bell stages, specialized structures like the enamel knot and Hertwig's epithelial root sheath play crucial roles. The enamel knot acts as a signaling center, guiding cusp, patterning, and tooth shape. Hertwig's sheath, derived from the enamel organ, determines root formation and shape. These structures coordinate the spatial and temporal expression of key signaling pathways, ensuring proper tooth morphogenesis. Their functions are now being mimicked in regenerative strategies to guide tissue patterning. Tooth morphogenesis is orchestrated by a network of signaling pathways, including BMPs, FGFs, SHH, WNTs, and TNFs. These pathways regulate cell proliferation, differentiation, and spatial organization. Misregulation can lead to developmental abnormalities. Recent advances in controlled release technologies now allow researchers to mimic these signaling patterns, offering new ways to direct tissue patterning and epithelial mesenchymal interactions in regenerative dentistry. Adult human stem cells are undifferentiated cells present in nearly every organ, capable of replacing damaged cells and supporting tissue renewal. They can be activated by injury or disease to proliferate and differentiate into tissue-specific lineages. In tooth regeneration, adult stem cells from dental tissues, bone marrow, and other sources are being explored for their ability to form dental pulp, dentin, and supporting structures. Their accessibility and multipotency make them attractive candidates for regenerative therapies. Dental tissues provide several types of stem cells with high regenerative potential. These include dental pulp stem cells, dental follicle stem cells from exfoliated deciduous teeth, apical papilla stem cells, periodontal ligament stem cells, and gingival stem cells. Dental stem cells are easily accessible, suitable for banking, and have high proliferative and multilineage differentiation potential. They are particularly relevant for dentin, pulp, and periodontal regeneration, making them central to current research. Dental stem cells are typically isolated through enzymatic digestion of tissue, followed by culture expansion in growth factor-rich media. For immature progenitors, multi-step explant culture is used. These cells exhibit fibroblastic morphology, adhere to plastic, and require specific media to maintain their proliferation and multipotency. Proper isolation and culture are essential to preserve their regenerative capabilities and ensure successful outcomes in tissue engineering. To confirm the identity and functionality of dental stem cells, researchers use in vitro assays like colony-forming unit assays, phenotypic assessment, and flow cytometry for surface markers. Multipotency is confirmed by directed differentiation into odontoblastic, osteogenic, chondrogenic, adipogenic, and neurogenic lineages. In vivo assays involve implanting cells into permissive hosts to observe survival, integration, and tissue formation. These tests ensure that only high-quality stem cells are used for regeneration. Prolonged in vitro expansion of stem cells can lead to decreased proliferation, telomere shortening, and cellular senescence, reducing their regenerative potential. To preserve stem cells for future use, cryopreservation in liquid nitrogen with cryoprotectants is employed. This method maintains marker expression and multilineage potential, allowing for later extraction and use. Proper storage and monitoring are crucial for maintaining the quality and effectiveness of dental stem cells. Scaffold-based approaches use 3D porous biomaterials to provide spatial cues, support cell interactions, and enable the transport of gases, nutrients, and regulatory factors. Ideal scaffolds are highly porous, biocompatible, and degrade in sync with new tissue formation. They must also match the mechanical strength of dental tissues and allow for vascularization. The choice of scaffold material and design is critical for successful tooth regeneration. Scaffolds can be classified by degradability, physical form, cellular content, origin, fabrication method, and function. Natural polymers like collagen, chitosan, alginate, hyaluronic acid, cellulose, and silk fibrin mimic the native environment and offer high biocompatibility. Synthetic polymers, such as PLA, PGA, PLGA, and PCL, provide tunable mechanical properties and degradation rates. Bioceramics and bioactive glasses mimic the mineral phase of hard tissues. Composite scaffolds combine the strengths of both natural and synthetic materials. Scaffold-free approaches rely on self-assembling cell constructs without exogenous biomaterials. These methods reduce the risk of immune reactions and scaffold degradation, preserve natural cell-cell and cell matrix interactions, and simplify clinical translation. Self-assembly is driven by cell adhesion molecules, while self-organization requires additional external cues for complex tissue patterning. Scaffold-free constructs can support vessel and nerve ingrowth, mimicking natural tissue development. Cell sheets are created using thermoresponsive polymers or ascorbic acid treatment to enhance extracellular matrix deposition. These sheets maintain intact cell-cell and cell matrix connections and can be stacked to form thicker, prevascularized 3D constructs. Cell sheets have shown success in regenerating vascularized pulp-like tissue and are straightforward to apply clinically, preserving the natural architecture of dental tissues. Cell spheroids are formed using techniques like pellet culture, hanging drop, and microwells. These spherical aggregates have a tripartite structure with an outer proliferative rim, a middle quiescent layer, and a necrotic core. Spheroids enhance cell viability, protein secretion, and differentiation potential due to their 3D architecture. They are especially useful for promoting vascularization and neurogenesis, making them ideal for pulp and dentin regeneration. Tissue strands are fabricated by injecting cell suspensions into tubular alginate capsules, which are then removed to leave scaffold-free, cylindrical cell-based strands. These constructs allow for linear arrangement of cells and integration of multiple cell types. They are ideal for bioprinting and can be customized for patient-specific pulp regeneration. Achieving mechanical stability remains a challenge, but tissue strands show promise for rapid, biomimetic tissue production. Vascularization is critical for the survival of engineered dental tissues, especially in the pulp cavity. Scaffold-free spheroids and strands enhance angiogenic potential through 3D architecture and cell signaling. Pre-vascularization strategies and the use of angiogenic growth factors are essential to overcome diffusion limits and ensure rapid blood vessel ingrowth. Without proper vascularization, constructs risk core necrosis and failure. Scaffold-free microstructures support the ingrowth of nerves, with dental pulp stem cell spheroids differentiating into neuron-like cells under specific conditions. These constructs also promote the formation of odontoblast-like cells that deposit dentin, aligning along the dentin surface. Proper innervation and dentin formation are vital for restoring the sensory and functional properties of regenerated teeth. Several factors influence the success of scaffold-free constructs, including diffusion and size constraints, cell number and density, vascularization and innervation strategies, and mechanical stability. Constructs larger than 1 centimeter require rapid vascularization, and cell density affects extracellular matrix deposition and signaling. Balancing mechanical strength with bioactivity and ensuring integration with host tissues are key challenges for clinical translation. Growth factors such as PDGF, TGF-beta, BMPs, VEGF, FGFs, IGF, NGF, and SDF orchestrate the proliferation, migration, differentiation, and organization of dental stem cells. Their effects depend on concentration, timing, and tissue location in regeneration. The goal is to reactivate these developmental cues to restore pulp, dentin, periodontal tissue, or even whole tooth structures. Controlled delivery of growth factors is a major focus in current research. Tooth development depends on the interaction of major signaling pathways, BMP, FGF, WNT, and SHH. BMP4 is crucial for the bud-to-cap transition. FGF8 and FGF9 initiate tooth formation, WNT signaling patterns, the dental epithelium, and SHH is essential for morphogenesis. These pathways are tightly regulated and interact with each other to ensure proper tooth shape, size, and function. Understanding and mimicking these pathways is key to successful regeneration. Modern regenerative strategies aim to recreate the developmental environment needed for tissue repair. Cell homing delivers growth factors into root canals to recruit endogenous stem cells, regenerating vascularized, innervated pulp-like tissue. Controlled release systems use biomaterial scaffolds to deliver growth factors in a sustained, targeted manner, avoiding burst release and preserving bioactivity. These approaches are at the forefront of clinical translation. 3D and 4D printing technologies enable the creation of structured scaffolds with precise architecture and programmable release patterns. These methods allow for the delivery of multiple growth factors at different times and locations, closely mimicking natural development. Personalized scaffolds can be designed to fit individual patients, improving the efficiency and effectiveness of regenerative treatments. Genetically modified cells, such as MSCs, dental pulp stem cells, and periodontal ligament cells, can be engineered to deliver regenerative signals directly at the site of repair. This approach allows for local production of cytokines and growth factors, potentially improving the activity and specificity of therapeutic molecules. Genetic engineering is opening new possibilities for precise, effective dental tissue regeneration. Whole tooth regeneration is the ultimate goal in regenerative dentistry. Bioengineered tooth germs created from epithelial and mesenchymal cells have successfully erupted and functioned in animal models, demonstrating the feasibility of this approach. However, achieving anatomically precise, vascularized, and innervated teeth suitable for human implantation remains a major challenge. Ongoing research is focused on overcoming these hurdles to make whole tooth regeneration a clinical reality. Biomaterials serve as temporary 3D extracellular matrices, providing physical support, guiding cellular attachment, and regulating the proliferation and differentiation of dental stem cells. They are classified into natural polymers, synthetic polymers, and bioceramics. Each type offers unique advantages, such as biocompatibility, tunable mechanical properties, and the ability to mimic the mineral phase of natural tissues. The choice of biomaterial is tailored to the specific tissue being regenerated. Different dental tissues require specific engineering strategies. For dental pulp, soft hydrogels and vascularization are prioritized. Dentin regeneration relies on hard, mineralized scaffolds, while periodontal ligament and alveolar bone regeneration use multiphasic or functionally graded scaffolds. These frameworks combine soft and hard materials to anchor teeth and integrate with surrounding bone, ensuring stability and function. 3D bioprinting allows for the precise placement of living cells and biomaterials, creating anatomically accurate tooth structures. Bioinks containing dental stem cells are printed layer by layer, replicating the geometry of a tooth crown or root. Smart scaffolds are functionalized to release signaling molecules in a controlled manner, coordinating multi-tissue healing. Nanocomposites enhance structural strength and accelerate stem cell differentiation, pushing the boundaries of regenerative dentistry. Regenerative dentistry is transforming clinical practice by using cells, growth factors, scaffolds, and advanced technologies to repair or replace teeth, gums, and jawbones. Minimally invasive treatments are now available for gum disease, early decay, and missing tooth structure. The field is moving from traditional drill and fill methods to biologic repair and remodeling, offering patients more natural and lasting solutions. Recent breakthroughs have enabled the regeneration of enamel-like material, dentin, and pulp using stem cells and growth factors. For example, stem cell therapy can regenerate dental pulp and dentin, preserving tooth vitality and function. Platelet concentrates and exosomes support healing and tissue formation. These advances are less invasive than traditional root canals and crowns, especially beneficial for young or immature teeth. Fully functional teeth have been grown in animal models using bioengineered tooth germs from epithelial and mesenchymal stem cells. These teeth develop roots, nerves, blood vessels, and integrate with the jawbone. Organoid technology and protein inhibition strategies are also showing promise. However, scaling these successes to humans remains a significant challenge due to differences in size, complexity, and developmental timing. Regenerative treatments are also making strides in bone and periodontal tissue repair. Bioactive materials and synthetic bone scaffolds stimulate new bone growth, improving outcomes for dental implants and treating severe gum disease. Mesenchymal stem cells and platelet-rich concentrates promote healing, while advanced scaffolds provide the necessary structure for tissue integration and stability. Acellular therapies using growth factors, extracellular vesicles, EVs, and platelet-rich plasma, PRP, or fibrin, PRF, are emerging as effective options. These factors orchestrate cell proliferation, migration, and differentiation, supporting rapid healing and tissue regeneration. EVs and exosomes carry rejuvenating signals, while PRP, PRF promote vascularization and osteogenesis. These therapies offer lower immunogenicity and tumor risk compared to cell-based approaches. Gene editing and organoid technologies are pushing the boundaries of regenerative dentistry. CRISPR and other gene editing tools can modify genes to support tissue regeneration and regulate inflammation. Organoids miniature, lab-grown tooth-like structures help researchers study development and test new therapies. These advances promise quicker recovery, personalized treatments, and steps toward functional whole tooth regeneration. Replicating the structure of enamel and dentin is a major challenge. Enamel is 96% mineralized hydroxyapatite formed only by ameloblasts during development. Once lost, it cannot naturally regenerate. Dentin formation depends on the spatial organization of odontoblasts and pulp. Without precise control, engineered tissues may calcify randomly, leading to non-functional or dead teeth. Overcoming these structural and biological hurdles is essential for true tooth regeneration. A promising alternative to cell scaffold engineering is the use of monoclonal antibodies like TRG-035, which target and block USAG1, a protein that inhibits tooth growth. By silencing USAG1, dormant tooth buds can be reactivated to grow new teeth. Early clinical trials show safety, but this approach only works if latent tooth buds are present. For many patients, especially the elderly, cell-based engineering remains necessary. Tooth regeneration raises important ethical and safety questions. Pluripotent stem cells carry a risk of tumor formation, and allogenic therapies may trigger immune rejection. Ensuring correct tooth shape, size, and integration is challenging. Using animal-derived materials in cell culture introduces contamination risks. Ethically, sourcing stem cells from naturally shed or extracted teeth is preferred, and transparent communication with patients is essential to manage expectations and ensure informed consent. Tooth regeneration stands at the intersection of developmental biology, stem cell science, biomaterial engineering, and clinical dentistry. Recent progress in partial regeneration is already informing clinical protocols, while whole tooth regeneration remains a bold but achievable goal. Overcoming challenges in cell sourcing, vascularization, morphogenesis, and integration will require interdisciplinary collaboration and continued innovation. With responsible research and clinical translation, the dream of regrowing natural, functional teeth is moving closer to reality.